The main challenge has to do with the availability of the testing materials. The machines can be available, but you might not have the reagents to be able to run on these machines. Even if the lab has the money to buy the reagents, it takes time. It takes a lot of time. It takes time to get to the country, it takes time for it to be cleared, it takes time for distribution. And most of these reagents have to maintain a cold chain. So if it takes long, you're not guaranteed whether it is kept at the temperature that it's supposed to be kept. And so the integrity of the reagents you are receiving is problematic.
We don't manufacture. We have to buy from outside of our countries. Even when we buy within Africa, say for instance, we buy it most of the time from South Africa. But South Africa don't produce either. They have to acquire from somewhere else.
Even at the African Union, they are now launching some initiative for local production, for reagent, for vaccine, to really help Africa as a continent to be more independent.
There are scientists in Africa who are capable, who have the capacity and the knowledge to be able to produce some of these reagents that we need for testing. The only challenge is facilities and equipment that you will need to produce this reagent. This is the biomanufacturing lab at ICGEB. So what I am trying to establish here is to establish a protocol that you don't need, just with a minimal pipette and pipette tips, you can use to produce some of the proteins and reagents that are needed. This should be an alternative to buy expensive reagents from other places. I think that if the African scientists can be empowered with some of these protocols and some of these techniques, then we can start meeting some of the local needs when it comes to especially proteins that are needed for nucleic acid testing in Africa.